So we now come to what is said to be the highlight of our annual meeting, but in fact I think we've had such a fantastic meeting that we'll just have to say that it's one of the additional speakers. And this is the Galton lecture, which has been given every year since 1914. And this year it's entitled Lionel Penrose and the Paternal Age Effect for Mutations, 60 years on. And I'm very pleased to welcome our speaker, Professor Andrew Wilkie, who's an FRS. Andrew Wilkie is the Nuffield Professor of Pathology at the Weatherhall Institute of Molecular Medicine in Oxford. And he trained both in Cambridge and in Oxford. And during his clinical year, he says that he was lucky to be sprinkled with some of the magic dust by Sir David Weatherhall. And we're very lucky to have Sir David Weatherhall as our last speaker this afternoon. He then took his Doctor of Medicine with Professor Doug Higgs in the alpha thalassemia mental retardation syndromes. And after completing his training as a clinical geneticist, he returned to Oxford in 1993 and set up his own lab, looking at the molecular genetics of congenital malformations. This led to his work, which we're going to hear today, and he's currently a Wellcome Trust senior investigator, and last year was elected to FRS. So, Andrew Wilkie, please come up and give your talk. Thank you. What you're supposed to do here. Do you know what you're supposed to do?
That's mine?
Yeah, great, thanks. Thank you.
Great. Well, thank you for that introduction, Philippa, and thanks very much for coming. So, yes, I think there are three ex-housemen of David's who've talked today. So I think that's a testament to the sort of amazing impact that he's had on academic medicine in this country. So we'll come on to Lionel Penrose in a moment, but actually the problem I'm going to talk about is one of the earliest, longest standing problems in human genetics. Goes back over a century to a study done by Willem Weinberg, the same man of Hardy-Weinberg equilibrium, on the inheritance of dwarfism. And so he was studying children, most of whom had achondroplasia. And what he noticed was that where the parents were unaffected of normal stature, the children with achondroplasia tended to be the last born of the sibship. And so what he wrote in this paper is if a more exact analysis of birth order indeed confirmed a high incidence in the last born children, this would point towards the formation of the anlage for dwarfism by mutation. Now, and this was a statement of sort of extraordinary prescience because it was more than 30 years before it was even known that the genetic material was DNA. But really the person who provided the real early insights into this problem was Lionel Penrose. So here is Lionel. This is a picture taken from the biographical memoirs of the Fellows of the Royal Society, which was written by his successor, Harry Harris, and as well as being a very distinguished, really polymath himself, he's had very distinguished offspring, and one of them is a friend and colleague, Shirley Hodgson. She's here today, just got back from India, so it's very nice to have you here, Shirley. So Penrose, actually, he did his first degree in moral sciences, but then switched to medicine. He became interested in psychiatric disease and mental disorder, and starting in 1930, began a long-standing study aimed at to increase existing knowledge in the causation of mental deficiency. And really based on that work, he was appointed in 1945 as the Galton Chair of Eugenics. And then really the key paper that he published that is relevant to the work I'm going to talk about today was in 1955, when he proposed that, in fact, this observation of achondroplasia, the key factor was paternal age rather than birth order. So a couple of things to just say about this. The first is that actually, although I consider this to be a classic paper, it wasn't even mentioned in the biographical memoir. Such was the breadth of what he did. And then the other thing that might surprise some of the younger members of the audience is to be a chair of eugenics, but of course Galton is the person who coined that term. And if you go onto the Wellcome Library's website, where you can find the Lionel Penrose papers, what you'll see is that actually, specifically, it says that he was a man of endless curiosity, succeeded Carl Pearson and R.A. Fisher as Galton Professor of Eugenics, a post he held from 1945 until his retirement. But then, however, he never liked the term eugenics. In 1954, he changed the title of the Galton Laboratories Journal from Annals of Eugenics to Annals of Human Genetics. And in 1963, he finally managed to get his own chair title changed. So, so he was actually ahead of his time in thinking in that way. But really the work that led to this work on achondroplasia, through his interest in mental handicap, of course, Down syndrome is one of the most common causes. And so he's very interested in the causes of Down syndrome. And in 1954, he published this paper in The Lancet. And one of the things he did in this paper was to look at the maternal ages of mothers who'd had children with Down syndrome that was running in families as opposed to sporadic Down syndrome. And he noticed that when it ran in families, there wasn't an elevated maternal age. And I suspect that this was one of the key things that made him think more generally about looking at age effects in sporadic disorders. And so this led to this further paper a year later, Parental Age and Mutation. And basically, his observation can be boiled down to this table here. And in it, he looked at the average difference between fathers' and mothers' ages. If you looked at a control population, it was 2.3 years. In Down syndrome, it was 1.7 years, so actually less than the average. And in achondroplasia, it was 3.4 years, so it's going in the opposite direction. Now, that might seem relatively small or subtle effect, but in his summary to this paper, this is what he wrote. It is assumed that gene mutations which are responsible for certain human diseases and malformations can originate in several different ways. An attempt is made to differentiate causes of mutation by examining their relationships to parental age. The influence of the father's age is shown to be of critical significance. When the effect of the father's age is appreciable, as in achondroplasia, the hypothesis of fresh gene mutation as the cause is strengthened. When it is absent, as in Mongolism, other mechanisms must be postulated. So, of course, this is incredibly prescient, because this is before we had any idea what was causing these things. But, of course, he was completely right. Down syndrome caused by trisomy 21. And then another sort of 40 years later, it's shown that achondroplasia caused by specific mutation in FGFR3. So you can now actually study what is the basis of these effects. And actually, the reason for the maternal age effect in Down syndrome still isn't fully understood. But what I hope to persuade you about today is we do understand more the basis of this. Now, if you go on to read the discussion of this paper, there's something else in it that interested me, because he writes, in other congenital defects, may sometimes arise as a result of fresh mutation, it is not unreasonable to search for evidence of relationship to parental age. An example is acrocephaly, considered by Ferryman to be due to mutation in many instances. A significant increase in maternal age has been found for this condition, Penrose, 1938. So acrocephaly is another word for craniosynostosis, all right? And so clearly what he was thinking at this stage, which remember is 1955, that this is something that shows a maternal age effect. So at this time, he's clearly interested in these age effects. So he recruits, as a PhD student into his lab, Eric Blank. And this nice picture of Eric has been kindly supplied by his family, Katrin and Catherine, and I hope they're here. Are you here? Hooray, you got here, well done. Because it was a bit of a struggle to get you tickets, but I'm delighted that you're here and welcome. So Lionel Penrose put Eric on to the study of the condition that particularly interests me, and I'm going to talk about more Apert syndrome. And this is his Eric's paper published in the Annals of Human Genetics in 1960. And so this is in the introduction, which really sets the scene. It was decided to reinvestigate the problems of definition and etiology to estimate the frequency of the condition. Of interest to was a possible parental age effect, suggested in Grebe's 1944 data and usually ascribed to an increase in the mother's age. So that's what we heard earlier. As Penrose, 1957b, pointed out, the age effect could be explained more satisfactorily as secondary to an increase in the father's age. So it's very interesting that in the 5 years between that original paper and now, clearly with his more sort of exacting analysis of the problem, Penrose has realised that actually this was a paternal age effect and not a maternal age effect. And then Blank goes on to describe the way that he did this quite by far the biggest study at the time of Apert syndrome in the UK. And of course this was 1960, so he very kindly and usefully provided in an appendix a list of the dates of birth of all these individuals as well as their parents, which obviously you wouldn't be able to do now for reasons of confidentiality, but we'll actually come on to this towards the end of my talk. So this is basically what Blank found in his study when he is looking at father's age, mother's age and birth order and comparing a control population, which is solid lines, with the Apert syndrome population, which is the dashed lines. And you can really appreciate the nature of the problem here because all these things there's clearly an effect for. So how do you disentangle those? So essentially with Penrose's guidance, Blank presents a method that uses partial correlations, which were originally described by Sewell Wright, and I don't pretend to completely understand how this is done. But the key conclusion is shown here, which is when you look at the correlation between the father's age and birth of Apert syndrome, holding the mother's age and the birth order constant, you still find a residual correlation which is significant in relation to the standard deviation, whereas it effectively goes away when you look at mother's age or birth order. So Blank's conclusion from this study was there is a marked parental age effect among sporadic cases of Apert syndrome. This is probably due solely to an increase in father's age. And actually we know now that he was 100% correct in that conclusion. So why would this matter? Why all this stuff about parental ages? Well, basically, the process of making eggs and making sperm is totally different. As most of you will know, if you're a woman, you've made all your eggs before you're even born, whereas if you're a man, you're making sperm through recurrent mitotic divisions throughout your reproductive lifetime. If mutation arises as a result of problems with replication, then the older you are as a man, the more replications your spermatogonial stem cells have been through, And therefore, you expect you're going to accumulate more and more mutations, as shown here. So basically, there's been this long-standing idea, copy-error hypothesis, that mutations accumulate through multiple replication errors in spermatogonial stem cells, and these increase in frequency with age. So this is correct, but what I want you to take away from my talk today is that when we're thinking about achondroplasia and Apert syndrome, there's an additional factor here which accentuates this, which we call selfish selection. Okay, so as Philippa introduced, when I was setting up my own lab and you're thinking, well, what do I work on? I chose to work on this condition, Apert syndrome, very characteristic combination of a craniofacial appearance caused by craniosynostosis and this syndactyly of the hands and feet. I was extremely influenced in this by going to a meeting arranged at EMBO in Heidelberg in 1990, just as I was finishing off my doctorate with Doug Higgs, the molecular biology of vertebrate development. And so this was a time when it was being learnt that the genes that were being pulled out of fruit flies and nematode worms, where actually you could find homologs in mammals, mice, and that these did important things in development. And really at that time, there was almost nothing known about what they did in human development. But a lot was known about the limb as a model system. And so my idea was that if you could think about the sort of molecules that could cause these limb malformations, then you could maybe learn, because you always get craniosynostosis in Apert syndrome, what causes the craniosynostosis, about which absolutely nothing was known. And in fact, half my lab still works on identifying genetic causes of craniosynostosis, which I won't talk about at all today. So at this stage, I teamed up with Sarah, as slaney as she was then now, Sarah Smithson, who's a clinical genetics colleague, to try and identify the cause of Apert syndrome. Now, one of the things about it almost always arises by new mutation, but it's not that infrequent, 1 in 65,000 births. And so that suggested to me that it was a big molecular target. So I won't go into how we found the mutations because there isn't time, but essentially what we found was completely the opposite, that actually the molecular target of mutations in Apert syndrome is exquisitely specific, whereby 99% of cases are caused by one or other of both C to G transversions occurring in adjacent codons in the fibroblast growth factor receptor type 2 gene. So remember, achondroplasia is FGFR3. So these are encoding specific amino acids at these positions, either serine to tryptophan or proline to arginine. And in some of the slides, you'll see the single letter designation, so that's S252W or P253R. Okay? So obviously this finding raised a lot of interesting questions, but the one for this talk that I've worked on ever since is, well, why is the mutation rate at these positions so astonishingly high? So to kind of understand that, you have to have a couple of further bits of background biology, and neither of these were work done by my group, but they're important for understanding the whole story. So the first thing is, this is a crystal structure of the FGF receptor 2 bound to an FGF ligand, because this is, as the name suggests, these receptors are involved in binding ligands. This was done by Musa Mohammadi in New York. And so this shows one of the Apert mutations, the substitution to tryptophan, and this shows the other one, the substitution to arginine. Both these mutations introduce additional contacts to the FGF ligand. So they're actually like souped up versions of the normal receptor that bind to the FGF more tightly than normal, so they gain a function, all right? Then the other question is, all right, so there's a gain of function, it's increasing the activity of the receptor, But there are a number of different pathways, intracellular pathways, through which that mechanism could be acting. And there's a lot of argument for the next decade about which pathways they were. And then Chung-Sha Deng did this really beautiful piece of work with an exact mouse model of Apert syndrome. So here's one of the mutations knocked into the mouse, and you can see it doesn't look very happy, and he's got praesynostosis. So this mouse in the middle is genetically the same as the one on the left, but phenotypically it's like the wild-type mouse on the right. So what's happened to this mouse? Well, when it was an embryo inside its mother's uterus, the mother was treated with a drug, and this drug, U0126, is a specific inhibitor of the RAS-MAP kinase pathway. We actually heard about this already this morning from Sadaf. So this is an example of pleiotropy where the same pathway is used in different cellular contexts. And so it inhibits MEK1 and MEK2 in this pathway and essentially abolishes all these phenotypic abnormalities. So that tells you that this pathway is critical in the manifestation of the craniosynostosis. And we'll come back to this pathway later on in the talk. So hopefully I've set the scene a bit. I've mentioned that achondroplasia is due to a mutation in FGFR3 and Apert syndrome to a mutation in FGFR2. These mutations actually locate in different portions of receptor. They're the most frequent transition and transversion mutations, respectively, in the entire human genome. That can't be a coincidence, surely. They're elevated but 500-fold over the background. So this is the big question, why? And although I won't say anything more about this achondroplasia mutation, I will mention a couple of other mutations in FGFR3 that are associated with more severe dwarfism phenotypes than achondroplasia, called thanatophoric dysplasia. So the first thing to ask if you want to work on this problem in any individual new mutation is, okay, did that mutation come from the mother or the father in this particular child? So to answer this question in Apert syndrome, we looked for nearby single nucleotide polymorphisms, and in those days there wasn't a catalogue to look up, you had to find them yourself. And fortunately there were a couple within shouting distance of the Apert mutations, and you can use these as markers for the two chromosomes, and in pedigrees that were suitably informative, like this one here where each of the parents is homozygous for the opposite allele, that provides in the child a marker for each allele. And then you can use allele-specific PCR to work out if the mutation arose from the mother's or the father's allele. And we got a very striking result, still I think the most extreme in any genetic disorder. Essentially, these mutations all came from the dad. And if subsequently there are a number of other studies of different mutations in FGFR2 and FGFR3, they always came from the father. People gave up doing these studies. So they're all associated with a paternal age effect here. And so apart from these two, there are three other gene disease combinations that are currently known with the collective properties having these high apparent germline point mutation rates, a very strong paternal bias of origin, and a strong paternal age effect. And these are HRAS, associated with Costello syndrome, PTPN11, Noonan syndrome, and RET, multiple endocrine neoplasia types 2A and 2B. So we've already seen a bit about the phenotypes associated with mutations in FGFR2 and FGFR3. These are the phenotypes associated with mutations in these other three genes, also associated with congenital disorders, but the other component that comes in here is that in each of these cases, these are known to be associated with a cancer predisposition as well, which isn't the case for these two genes. Again, this turns out to be significant. So we've established that these come from the father. So you've got to try and measure these mutations in the sperm. And although it was relatively easy to establish the paternal origin, it took another 7 years before we were able to design an assay to actually measure the level of the APIT mutation in the sperm. And really, I made no progress until I hired my secret weapon, Anne Gorielli, who's sitting in the front row there, who did a beautiful piece of work on this. So I'll go through this in some detail because this is quite crucial to the story, really. So what this shows is the level of this APIT mutation. It's the 755 C to G mutation, measured in a number of different samples. This is the age on the x-axis, and this is the mutation level on the y-axis. And this is 1 in a million here, and it's on a log scale. Okay, so the limit of our detection using this assay is about 1 in a million. So if you take a blood sample from someone and you try and find this APIT mutation, well, you can't. It's less than 1 in a million. This is not something that's in the blood. All right. If you take semen samples, these are just from normal, healthy male donors in the population. There's some who aren't producing it, but the majority are producing it in their sperm. And the highest level we've seen is about 1 in 6,500. So this is something that men are doing. There's a strong correlation with the age of the man. And then the 6 turquoise diamonds are from analyses of semen samples from men who've actually fathered a child with this particular APIT mutation. And what you can see is that their values, although they're at the higher end of the normal range, they fall within the normal envelope. So there's nothing different about a man who's fathered a child with APIT syndrome from anyone else in the population. This is something that men do. And if you take these data and put them into 5-year bins, you see it goes up with age. And if you relate that to births of children with APIT syndrome, it correlates very closely. So this explains the age effect, but it doesn't, of course, explain why these levels are so high in the first place. So to think about that, let's go back to our SNP that I mentioned before and think of 2 alternative extreme hypotheses. One is that this is some sort of intrinsic mutation hotspot that's just recurrently mutating. And then all things being equal, there should be an equal distribution of mutations on either FGFR2 allele. So when you look at the mutant alleles, they should distribute equally across the 2 alleles of the SNP. The opposite possibility is actually the mutations themselves are rare, but once they occur, they're subject to some positive selection. And if the mutations occurred only once, it can, by definition, only be associated with one allele of the SNP, and then you'll see 100% distribution, which could be either allele. So these are the data that we got. So this is the proportion of the mutants that are on the G allele of the SNP. So you can see, and this is the level of the mutation. So less than 1 in a million we can't measure, so they're around 50%. They've got massive error bars, so that means nothing. But where there are enough mutant molecules that we could measure this allelic skewing, far from being a 50-50 distribution in most cases, there's actually very marked skewing towards one or other allele, but there's no preference for one or the other. So really this led to our formulating the hypothesis that although the mutation obviously has to occur in the first place for this to happen, really the key factor here was selection, positive selection occurring after the mutation had arisen, remembering that these are gain-of-function mutations. We've called this selfish spermatogonial selection. That mutation is originally only in one cell, so it takes time for clonal expansion to occur, and this is the basis of the age effect. So imagine a mutation arising at this stage on the green allele and gradually expanding with time. Here, a second mutation occurs that happens to be on the other allele, but it can never catch up with the first one. Right? And so that is part of the basis of this skewing effect. So really the next sort of development in this was through a group in Los Angeles led by Norman Arnheim, who had the idea of cutting up a testis into about 200 pieces. So these were from cadaveric donor banks. registering the position of each piece in the testis, developing a method, and they used a different method from ours, to measure the level of APETS mutation in a testis piece, and then construct a sort of heat map. So these are 5 testes reconstructed using this method to see where the mutations lay. And again, as predicted by our work, rather than there being a sort of uniformly moderately high level of mutations, what they found were these hotspots of mutations surrounded by large deserts. So we've got the idea that these are gain-of-function mutations associated with some sort of selective advantage and growth over time. So then really the next sort of piece of the jigsaw came in 2007, 2008, because I've said that there's been these aren't known cancer predisposing genes, all right, FGFR2 and FGFR3. However, if you look at endometrial carcinomas, one of the commonest mutations you find as a somatic mutation in endometrial carcinoma is in FGFR2. And if you look at what mutations you find in FGFR2, the APET mutation is the most frequent mutation that you find in FGFR2. So these are indeed, in the sort of classic somatic oncogenesis, these are oncogenic mutations. So this is our model at this stage. So we've got spermatogonial stem cells, which have a fine balance between needing to proliferate and self-renew and differentiate and make sperm, and that a mutation such as an APET mutation would slightly alter this balance in favour of proliferation. So you get an expanding clone of cells over time. So this clearly has close parallels with oncogenesis. And so this then made us think, well, could you actually find these kinds of mutations in testicular tumours? And the common types of testicular cancer affect younger men, usually between the age of 25 and 35, so they really don't show the right epidemiology. And so we teamed up with Eva Rajput and mates in Copenhagen, who'd collected really probably the largest number in the world of this rarer type of testicular tumour called a spermatocytic seminoma, which affects older men. So I set a DPhil student, Ruth Hanson, onto looking for paternal age effect mutations in these tumours. And of course, she started off with my favourite APET mutations. However hard she tried, she could not find any FGFR2 mutations in them. So this is when you have to kind of go off your favourite gene and say, well, let's look a bit further. And so Ruth started looking at the other paternal age effect genes. And she came into my office one day sort of brandishing these electropherograms. And in each of these cases, the top trace shows normal tissue, and then the other traces are tumour tissue from the same patient. And at this position here, there was an A to G substitution. So this made me very excited because in the germline, this substitution in FGFR3 causes type 2 thanatophoric dysplasia. So Ruth and then subsequently Indira Taylor went on and looked at the other 3 paternal age effect genes. And it turns out that the most frequently mutated gene in spermatocytic seminomas is actually HRAS, and so we found 3 different gain-of-function HRAS mutations in spermatocytic seminomas. And what this illustrates is that the tumours in which you find mutations are actually tend to be taken from the older men, so implying that actually this tumour itself is genetically heterogeneous, and what we're defining is an older age subtype, which hadn't been realised before. So at this stage, how do we tie all this together? Well, actually, these 5 paternal age effect mutation genes, the classic genes, all fit into a single pathway, and it's one we've heard about before, because it's a pathway where you signal through receptor tyrosine kinases, and then through RAS down to MEK and ERK. Okay, so they all fit into this single pathway. And so these are obviously involved in many different cellular processes depending on context, proliferation, self-renewal, survival. And you can speculate that any gene involved in controlling these processes could be subject to this effect. And so one pathway that immediately caught our interest is the PI3 kinase AKT pathway, because it's also a well-known pathway, particularly in survival. And there's close interaction between these two pathways. And there's some very nice work in the mouse done by Shinohara's group, where they've looked at this. And so this graph here shows the culture of mouse spermatogonial stem cells. And if you look at the red bars, which are wild type, to culture these cells normally, you need a cocktail of growth factors. So there's epidermal growth factor plus fibroblast growth factor plus glial-derived neurotrophic growth factor. Okay, so you need a cocktail. And if you take away any of those, then they don't grow. So if you transfect these cells with the gain-of-function G12V mutation in HRAS, they become completely growth factor independent. Okay, you can then look at the pathways that are signaling downstream of that using various inhibitors. So this is a pathway of the RAS-MAP kinase pathway, and this is inhibitors of the PI3K AKT pathway. And so in the wild-type situation, actually this amplification effect is driven by PI3K AKT. And with the mutants, with the G12V, it's a combination of both PI3K AKT and the RAS-MAP kinase pathway. And you can take these transfected cells and reimplant them into mice and get both regions of the testis that are able to undergo spermatogenesis and other regions that form tumours. So these are all the processes that are very much parallel to the ones we think we're seeing in the human. So then the next question, which we started in about 2008, was to say, okay, can we actually see this process occurring in the normal testis? And at this stage, another long-suffering DPhil student, Jasmine Lim, joined the lab, and so I set her on to working on this. And that, the thing is that although there are people who have their professions looking at testes every day, no one had actually seen anything like this happening in normal testes. So using our ideas about spermatocytic seminoma, and the fact there's certain immunohistochemical markers which are expressed in those tumours, we looked for a marker to screen testes with. And the one that Jasmine came up with was this one, MAJ4. So here's a cross-section through a number of seminiferous tubules. Each of these would be about 200 microns across. And this MAJ4 is marking the spermatogonial stem cells which are lying along the basal lamina, and then as they differentiate, they move towards the lumen and then release the mature sperm in the lumen. So then Jeff Mayer joined the lab, and what he noticed using this marker and sort of stepping back and looking at testis sections under low power is that some tubules seem to show more intense staining with this MAJ4 marker in a kind of double ring. And here's a close-up of part of this where you can see there are some tubules which seem to be staining more intensely. So that's interesting. And so the next thing you do is say, well, if you follow it through sections, does it look reproducible? If you use other antibodies, do you see similar effects? So this is a series of sections using both MAJ4 and a number of other antibodies where this seems to be something consistent about particular tubules, and they also stain positively for other significant antibodies. This is one to FGFR3, and this is one to phospho-AKT, all right? So these tubules seem to be different from their neighbours. Remember, this is just from normal testis. you seem to see clusters of them. So then, of course, the question is, well, do they join up or not? So then Jeff did very painstaking sections through literally hundreds of sections of these, sort of colouring them up, and then labelling them and reconstructing them, which he did with Steve Taylor. And indeed, all these join up. So they're a single contiguous tubule. These can go on for millimetres or even up to a centimetre. So this is the stage of the published data. So what I'm going to tell you about now is unpublished. Obviously, the question is, can we find mutations in these tubules? So what Jeff has done is to take tubules with appearances like this and micro-dissect them. Remember, this is formalin-fixed, paraffin-embedded tissue. There's not very much of it. It's very poor quality DNA. You have to whole genome amplify it, and then use a targeted resequencing approach using haloplex capture, and then a panel of our sort of favourite genes, somewhere between 100 and 150. And he's applied this to a number of testis sections. So what do you get? Well, the answer is that largely you get total garbage, actually, because this whole genome amplification of these very poor quality FFPE samples just generates masses of artifacts. So this is a comparison of the number of unique variants if you compare frozen tissue with whole genome amplified FFPE tissue. And by definition, these have got to be almost all artifacts. However, in one of the first testes he worked on, and this is from a 75-year-old man, it was removed because of a cyst. What Jeff noticed was that among 144 of these variants, there was something that he recognised. Sounded quite interesting. And so what he's able to do then is go back to an adjacent section, micro-dissect that adjacent section, and then analyse it by PCR and find out if that mutation's actually there in the adjacent section. And indeed, this mutation here was present in the adjacent section that stained abnormally, but not in a nearby section that had normal appearances. And this mutation is one that in the germline causes type 1 thanatophoric dysplasia. Having established a pattern like that, what Jeff can do is then take a sort of bigger region of testis where you can identify tubules that seem to have this different, more intense staining, and there's actually a perfect correlation between the ones that have this abnormal appearance and the presence of the mutation, which clearly indicates that these are all part of a single contiguous tubule that's moving in and out of the plane of section. So this is all great, but I was rather upset because this story's rather deviated from APET mutations for quite a while, since about 2007 really. So then, and this is the 8th of October, so this is really, this is less than a month ago, I get an email from Jeff. It looks like we've spoken too soon about not finding common PAE mutations in immunopositive tubules. So here are a couple of sections. This is actually from a 40-year-old man who had his testis removed because of a hernia. And this section here seemed to have this unusual appearances. So what Jeff did was to, we're now, because of this artifact approach, what he's doing now is to take 3 adjacent sections, amplify them independently, and then ask what's the overlap between the variants that you find in all 3 independent sections, and compare it with a final section where you scraped all the tubules off and amplified that to provide a control. And so what Jeff had found was that in these 3 adjacent sections, there's this mutation in all 3 that wasn't present in the whole section, and this encodes the proline to arginine mutation in APET syndrome. And so what I was even more pleased about was that if you look at the date here, this date here, 2nd of October 1994, and this is from my lab book. And these restriction digests were the first time that I was able to confirm the presence of this mutation. You know, from having read a sequence trace, this was really the key piece of evidence. So it took nearly exactly 20 years to go from this to go to this, which is quite satisfying for me. So some people can say, well, you know, this is all human. What about, why don't you work on animals? And we've always thought, actually, that here, the human's the best model organism to study this particular problem. However, we're very pleased to see that Marco Saindel, who's at Cornell, just a couple of months ago, published the first study where he's looked at this process in the testis. Now, even more pleased to say that he's chosen one of the APET mutations as his model. And essentially what he's done is to label up wild-type FGFR2 and APET mutant FGFR2, two different colours, and then look at competition between these both in vitro and in vivo. And he's got some evidence from that to support the idea that the mutant cells can outcompete the wild-type cells in vivo. So they populate the testis to a greater extent. So something that actually relates to what Naz was talking about this morning and cancer predisposition genes is I've been talking about all these links of FGFR2 with, you know, selective advantage and somatic mutations. So how can we be sure that FGFR2 isn't a cancer predisposition gene? And specifically, and important for me, are people with APET syndrome prone to cancer? And the problem is that we see these individuals as children in the craniofacial clinic, and we follow them up until they're about 18 and they're fully grown, and then they're discharged. They go out into the community. No one sees them again. No one knows what happens to them. So no one has any data on this. But here we come back to Eric Blank's study. Because just as Eric was retiring, my colleague Sarah got in touch with him, and he still had the records of these individuals, including their names. And so we got ethics permission to set up a study with, as it was then, the Office of Population Censuses and Surveys, to find out what had happened to these people. So this was first of all done in 1995, but in fact they've been followed up ever since. This has never been published. But just last year I sort of decided to finish the study and kind of break the code. And so this is the data. It's the first time it's been presented. And so these show the Kaplan-Meier survival curves for these individuals that Eric ascertained in 1959. And about a third of them are still alive now. And what you can see is that there has been some cancer-related mortality, but in fact, if you do the statistics as far as I can do them, I'm not an epidemiologist, this falls within the normal range. So there's not some strong indication that many of these individuals are going to suffer from cancers in middle age, which is really good news. So I'm really sort of coming to the conclusion. So we've heard a lot today about somatic mutations and germline mutations. So one of the concepts you can take away from this is you imagine a somatic mutation and you move it down to the testis. Then in this unique environment, it can behave in both ways. It can behave as a somatic mutation, giving rise to a selfish clone or rarely a testicular tumour, or if a sperm from one of these clones fertilizes the egg to produce an individual in the next generation, it can also cause a germline disorder. And there's an interesting conflict here, because these mutations are actually beneficial in the context of the testis, yet they're harmful to the organism. And so if we think about how this can play out, you imagine you have two different, remember these mutations themselves, they're totally random. Here you have a mutation that's not associated with any gain of function, and here's one that's associated with a strong gain of function. So they start off equivalent, but over the course of time, because of the gain of function, this one increases in number, there are more sperms with it. But then, because it causes a serious disorder, there isn't any transmission to the next generation. But you can do the kind of thought experiment where a slightly less activating mutation that's associated with a slightly smaller clone, not so much spermatogonial enrichment, could be associated with complex disorders such as cancer or neuropsychiatric disorders, or a lesser amount might be associated with susceptibility variants. And that these things, rather than stopping each generation, could actually be transmitted to the next generation and actually be fueling evolution and fueling the nature of our germlines. So, in a sense, the question at the moment is, there's a lot of dark matter out there. This is a log scale of the background mutation rate, and this is the background mutation rate in our genome for different kinds of nucleotide substitution, between 10 to the minus 7 and 10 to the minus 9. The sort of things I've been talking about are out here. They're way off the scale. They're sort of 500 to 1,000 folds more frequent. So what lies between? We don't know. And depending on whether the spectrum is like this, where there are any tens of mutations subject to these phenomena, or whether it's like this, it's going to make a big difference to what the overall impact of this kind of phenomenon is to disease overall. Does this matter? Well, this is a plot of the average age of paternity in England and Wales from 1950 to 2010, and it's actually gone up by 5 years in that period. So now over a third of babies are born to fathers over 35 years old. And if you believe the New Scientist, which published an article about this earlier this year, this is a testicular time bomb. This rather nice illustration here too. So what things going to be like in another 60 years? We've got these three wise men looking down on us, and one thing is going to be sure, I won't be around. But I think we can be sure about some things, that there'll be a comprehensive catalogue of mutations that are positively selected in the testis. And we'll have a broader view of the consequences for human evolution and disease. I think it's really certain that maternal plasma screening of cell-free fetal DNA, which is being applied successfully to aneuploidy, as sequencing techniques become more accurate, these paternal age effect mutations will be the first ones to which they are applied. But beyond that, I'm not a very imaginative person, so I'm not very good at seeing into the future. So the sort of thing you can think about, which to me seems impossible, is could you actually intervene pharmacologically to stop these gain-of-function mutations increasing in the testis, so that you could actually prevent this happening in the first place? Now, that seems highly unlikely to me, but as I say, that's probably just reflects my lack of imagination. So finally, I'd just like to thank the members of the Wilkie team who've done this work, and it's been brilliantly led over the years by Anne, and then the other members who've contributed to this have been Eleni and Indira and Jeff, and the other people work on craniosynostosis. Of course, this wouldn't be possible, first of all, without very long-standing funding from the Wellcome Trust, whom I'm particularly grateful, and a large number of collaborators. I'm not going to read out everyone's names, but I would particularly like to mention Gil McVean, who's given us fantastic statistical genetic support, and Eva, who's collaborated with us on the spermatocytic seminomas, and the Oxford Centre for Histopathology Research, which has supplied us with these testis samples. And with that, I'll finish, and thank you for listening.